Welcome back, students. In this part of the video, we will be learning about meiosis. Now, meiosis occurs in germ cells, that is the reproductive cells. Meiosis has two steps, that is meiosis 1 and meiosis 2. So, the cell division takes place in two steps. One cell only is divided into two steps. First is it will divide itself into meiosis 1, and then again whatever is the result of the first division, those cells again are divided in meiosis 2, and whatever is the final result, that is our product that we get from meiosis. So in meiosis, single cell, it divides in two steps, meiosis 1 and meiosis 2, and it produces four cells. So from one cell we get four cells. This meiosis, it occurs in our germ cell, that is the reproductive cells. The chromosome number first, it doubles up. So whatever the chromosome number, for example, the chromosome number is 2n. So it doubles up into 4n. And from 4n, it will give 2n and 2n. So our result becomes 2n and 2n, and from these 2n, 2n, these again divide and they form 4n number of chromosome cells. So this is how meiosis takes place. Now meiosis has, first the meiosis 1 has prophase 1, metaphase 1, anaphase 1 and telophase 1. Then meiosis 2 has prophase 2, metaphase 2, anaphase 2, telophase 2, and lastly our daughter cells are produced. Let us first start with meiosis 1. Now meiosis 1 first has prophase, which includes 5 phases. Then we have metaphase 1, anaphase 1 and telophase 1. So first let us study the 5 phases of prophase. So these prophase, these play a very important part in the cell division. Most of the work is done in the prophase 1. So let us first see what are the steps or the phases in prophase 1. Prophase 1 has 5 phases or the 5 steps. First is leptotene, zygotene, pachytene, diplotene and dikinesis. Now, prophase 1 is not visible with our normal microscope, and to observe prophase 1 we require an electron microscope. Also, most of the major events of the division in meiosis takes place in prophase 1. Let us first look at the leptotene phase. Now before the leptotene, the cell will divide itself, that is we have already studied about the interphase in which it is the resting phase and the cell prepares itself for the cell division. First phase is the leptotene. Now in this, condensation of the chromosomes takes place. You can see already the chromosomes are replicated. Here the condensation takes place. And also you can see that two centrioles are present here. Nuclear membrane has not started to divide, only the chromosomes have got condensed and it is easily visible to us. Next step is zygotene. Now zygotene is an important step in this. Synapses between the homologous chromosome occurs. So homologous chromosomes, it goes into a process that is called as synapses. Now synapses is the pairing of the homologous chromosome. So the homologous chromosome starts to pair each other. Now what are homologous chromosome? Chromosome of the same origin. That is from the mother and the father, from both which are of same origin, they starts to come together. Next step is pachytene. Now here a very important step takes place, that is in pachytene the crossing over takes place. Now what is crossing over? That is the chromosomes, they come along each other and they cross each other. Now why crossing over is done is so that the genetic material, it can be recombined. Now this is also called as the recombination, so that the genetic materials recombine. It will overlap each other. And also you can see that from the centriole, the spindle fibers have started to arise. The nuclear membrane has not started to disappear now. Only the crossing over is taking place, that is called as the recombination of the chromosome takes place. Here you can see the crossing over is being done. One chromosome, the ends of the chromosome are crossing each other. ये क्यों हो रहा है? So that the genetic materials recombine. जितने भी genetic material एक दूसरे में है वो सारे एक दूसरे के पास चले जाए, उसके लिए this recombination takes place. Next is diplotin. Now after the recombination, the chromosome it starts to separate each other. Now as it is separating, ये देखो यहाँ पे ये separate हो रहा है. जैसे जैसे ये separate होगा, इसका जो part है ये वाले क्रोमोसोम का, it will go into ये वाला क्रोमोसोम, एंड इसका जो थोड़ा सा पार्ट है इट विल कम इनटू दिस वाला क्रोमोसोम. सो जैसे वो सेपरेट होगा, एक दूसरे का जो जेनेटिक मटेरियल है वो भी एक दूसरे के अंदर चला जाएगा. सो इट इज स्टार्टिंग टू सेपरेट ईच अदर. एंड ऑल्सो इन दिस स्टेप द न्यूक्लियर मेम्ब्रेन जो है इट स्टार्ट्स टू डिसअपियर. So in diplotin the nuclear membrane is starting to disappear, and also the chromosomes are going away from each other, and as they are going away, along with it they are taking the genetic material of each chromosome. दोनों ने एक दूसरे को combine कर लिया. इसके पास इसका क्रोमोसोम, इसके पास इसका इसके पास इसका genetic material, इसके पास इसका genetic material. Next we have dikinesis. This is the final stage, and in this stage you can see the spindle fibers have arisen, the chromosomes are almost away from each other. See here, the chromosomes are almost away from each other, and in this phase the spindle fibers also starts to arise and it goes from the centromere. So this is the 5 phases of prophase 1. First was liptotin जिसमें कंडेंसेशन हुआ डीएनए का. देन वॉज जाइगोटीन जहाँ पे जो होमोलोगस क्रोमोसोम थे, सेम क्रोमोसोम्स थे, वो एक दूसरे के पास आने लगे. देन वी हैव पैचेटीन. इन पैचेटीन द क्रॉसिंग ओवर टुक प्लेस. Homologous chromosome they start to overlap each other, that is called as the crossing over. Then we had diplotin. In diplotin the homologous chromosomes they starts to repel each other, and along with the repelling वो लोग ने एक दूसरे का genetic material भी share कर दिया. And in dikinesis then the division is starting, that is the spindle fibers is arising, it starts to go from the centromere, and then in the next phase it will start to divide. So these were the 5 phases of prophase 1. Leptotin, zygotin, patchotin, diplotin and dikinesis. So now after the 5 phases of prophase 1 comes metaphase 1. Now in prophase 1 first was leptotin, zygotin, patchotin, diplotin and dikinesis. In dikinesis the spindle fibers has arisen, and their spindle fiber is passing through the centromere of these particular chromosomes, and also you can see the recombination has taken place, the chromosomes have recombined. Next step is metaphase 1. Now these are similar to as mitosis. The same things happen here also. इसमें भी the spindle fibers have arisen. ये जो all the chromosomes are there, they start to come in an equatorial plane and they starts to be in one plane, that is they start to arrange itself in the middle. ये देखो यहाँ पे they have arranged itself in the middle. Next is anaphase 1. They get pulled from each other, the centromeres break, and जितने भी क्रोमोसोम थे यहाँ पे आधे चले गए, यहाँ पे आधे चले गए, यहाँ पे आधे चले गए, यहाँ पे आधे चले गए. So from here the anaphase 1, where the chromosomes are being pulled away and like a bunch it appears like a banana bunch. Then next we have telophase. In telophase the nuclear membrane it starts to reappear. Now the cell has been completely divided, notch होने के बाद में, and from one cell we have got 2 cells. अभी you will see it in the color combination, both the chromosomes have mixture of both the cell. Both parents ka chromosomes are there in one particular thing, that is half blue and red is also there. Blue, red, you can find everywhere it is there. So that means the mixture of all the genetic material is done. So this is how prophase 1, and after that metaphase 1, anaphase 1 and telophase 1 is done in meiosis 1. So from meiosis 1, from one particular cell we got 2 different daughter cells. So one parent cell we got 2 daughter cells from telophase 1. सो मियोसिस 1 में एक सेल में से हम लोग को 2 डॉटर सेल मिलते हैं. सो फ्रॉम वन पेरेंट सेल वी गॉट 2 डॉटर सेल्स. एंड हियर यू कैन सी इन टेलोफेज 1 जो 2 डॉटर सेल्स है इट आर कंप्लीटली अवेलेबल एंड दे हैव ऑल द थिंग्स विच आर देयर. नाउ अब आगे जो होगा मियोसिस 2 में ये जो 2 सेल्स है दिस विल देन अगेन डिवाइड एंड इट विल form more number of cells. After meiosis 1 we have meiosis 2. So the parent cell got divided and 2 daughter cells were formed in meiosis 1. In meiosis 2, वो जो 2 daughter cells were formed in meiosis 1, those will undergo division and they will form 4 daughter cells. So from these 2 daughter cells, we will get 4 daughter cells. The one will divide and form 2, this other one will divide and form 2. The same process takes place, that is the cell undergoes after the prophase 2, it undergoes into the metaphase 2. That is the spindle fiber starts to arise. These chromosomes they starts to align in an equatorial plane. In anaphase 2 these are pulled away from each other. From the centromere they are pulled away and a notch is being developed in anaphase 2. And in telophase 2 the cells are completely divided, the nuclear membrane has appeared and these chromosomes have also divided and they have formed 4 daughter cells. Now if you will see, all of them have the genetic material same as the parent cell. So in meiosis, from one cell we get 4 cells. And also one thing you have to remember, that meiosis only occurs in the germ cell, that is your reproductive cell mein hi meiosis will takes place. So students, in meiosis, first we had a parent cell. From the parent cell, through meiosis 1, we got 2 daughter cells. That is 2 daughter nuclei, or you can call it as 2 daughter cells. And then after meiosis 1, now we had studied the steps also. In meiosis 1, you have prophase 1. Prophase 1 mein you have liptotene, you have zygotene, pachytene, diplotin and dikinesis. Then you have metaphase 2, anaphase 2 and telophase 2. In meiosis 2, वो जो 2 daughter cells are formed, it again goes into the same process. पूरा जो meiosis का step है, that is first it will go into prophase 2. But now they don't have liptotene, zygotene and all. खाली it will go into prophase 2, then metaphase 2, anaphase 2 and telophase 2. And we get 4 daughter nuclei at the end. So from one parent cell, we are getting 4 daughter nuclei. That is 4 gametes are being formed. And it occurs only in the germ cells. With this, we complete our second chapter in Science 2, that is Life Process in Living Organism, Part 1.